leading place for metabolic research. Then she did another kind of postdoc in the Netherlands at Maastricht University. And then she joined the faculty at the Translational Research Institute for Metabolism and Diabetes in Orlando, and the faculty at Sanford Burnham Research Institute, and which is a leading place in metabolic research, and particularly how it relates to translational research. And Dr. Spark is really a rising star, already a star in many things that are of interest to our group. While she's telling me she hasn't really done much aging research per se, I'm trying to convert her so she can apply her as I was converted some years ago, her expertise into the aging field. But she's really someone that is a leader in many important topics that are relevant to us: mitochondrial biology, adipose biology, muscle biology, exercise. So all these things that are so important for aging research and metabolic research. So I know she's been doing very exciting research in recent years, and I was hoping that she would share that with us.
Definitely.
Okay, thank you.
Thank you. Very nice introduction. And just to double check, everyone can still hear me? Okay, great. Yes, thank y'all for having me. I know I've talked to many of you about this, but you've endured the spillover from Harvey with the gas shortage, but I can relate because I will be getting on a plane tomorrow to fly into Irma, which most people, I think, are probably trying to get out of the path of Irma. But I'm from South Louisiana, so not my first rodeo with hurricanes. So just trying to stay calm. So thank you for having me. I'd like to show a picture of exactly where we are in Orlando and notice the absence of Mickey Mouse and Disney, because this was one of I had great pause when I was asked to join the faculty there, because I thought, oh, I don't want to move to a tourist trap. I don't want to deal with that. Luckily, Disney is 45 minutes away in Kissimmee. They like to say Orlando, but it's in another city all its own. So actually, let's see. There it is. So this is, so Florida Hospital is the largest nonprofit hospital system you've probably never heard of. It's part of Adventist Health System. They have about 10 campuses in the state of Florida. Their headquarters are now there. And this is their flagship hospital. It's a 2,200-bed hospital, and that's the campus that we're on. It's near downtown. So this is the Diabetes Institute right next to it. This is our nice brand new little institute. I think it opened up in March of 2012. So we're still pretty new. And then this is Sanford Burnham. It's about 20 miles down the road in Lake Nona, but it's all still considered Orlando proper. So just to kind of orient you geographically. But as I'm sure you saw from the title, I'm here to talk about what I call non-response. And so I'm interested in non-response, of course, to exercise, but this is not anything new or specific just to exercise. So what I mean by non-response is this physiological non-response that is, of course, driven by a person's genetic code that then is dependent on how things are transcribed, translated, post-translationally modified. In response to some sort of an intervention or, for instance, pharmacotherapy. So drug resistance can be an archetype for this non-response. And many of you may know this, but it's a beautiful linkage between physiology and genetics, because there are people that are based only on a single mutation, thank you, in certain genes. They are either a rapid or a slow metabolizer of drugs. So you can imagine this becomes very important. That's hysterical. This becomes very important. Yeah, I know. Leaning Tower of Pisa, let's see. So this becomes critical, as you can imagine, especially for, cancer drugs and things like that. If someone's going to metabolize a drug rapidly, then there's no point in prescribing that drug. And there are other examples, but a simple DNA test now can identify these risk populations beforehand, and you can just simply put them on another drug. So it sort of sets this precedence for precision or personalized medicine using someone's genetics. And it's not specific to drug resistance. This has also been shown with weight loss interventions. It's something else that I certainly did during my PhD and continue to do. And the NIH actually convened a workshop of the leading experts a couple of years ago, and they issued this report where basically it's a guide to weight loss success using a person's genomic information. So again, setting this precedent for these two fields are already ahead of us, us being the exercise field, in terms of personalizing a prescription. So that's my goal, the goal of my research is to understand why certain people don't respond to exercise, and to hopefully move us toward this sort of shifting the paradigm to be able to target treatments to people that don't respond, and first identify them and then tailor those treatments. And I'm focusing this on genetics, epigenetics as my sort of as my research focus. But a disclaimer, whenever I say non-response, people say, oh, that's not true. But what I mean by that is a non-response in one outcome does not mean a non-response in all outcomes. So it certainly matters what outcome you're talking about. So for weight loss, it's obvious the outcome is weight loss. But for exercise has head-to-toe benefits, as we all know. So you need to be specific about the non-response, and it's going to depend on that particular person and their health goals. So for someone with type 2 diabetes, it's going to be insulin sensitivity, glycemic control, and at the heart of that, or what's tied to it, is mitochondrial function, which I think is something all of us in this room can at least agree on. So what can influence this response or lack of a response, or at least the heterogeneity? And you have endogenous. I like to divide it up between endogenous and exogenous factors. So endogenous meaning not attributable to any external or environmental factor. I Googled that, so feel free to do the same. But these are basically things that are intrinsic that you can't change. So your age, sex, family history of diabetes, genetics, epigenetics. I'm sure there are others. So for this audience, I know you'll care about aging, and aging has been associated with an impaired hypertrophic response to resistance exercise training, so weight training. It's been termed anabolic resistance. But actually, a recent paper by Sri Nair and his group at the Mayo showed that there were equal adaptations to high-intensity aerobic training in terms of insulin sensitivity, skeletal muscle, mitochondrial function, in both the younger and the older individuals that were trained. So it kind of argues a bit against that age being sort of the driving force of a non-response, at least in terms of insulin sensitivity and mitochondrial function. There's also data from Francesca Amati that's shown that these chronically trained older adults actually have high mitochondrial content, function, and exercise efficiency compared to sort of their younger athletically trained counterparts. So the bottom line here is that aging in general has not been thoroughly examined in this context of non-response, where you're identifying this subset of a population that doesn't respond. So I will leave that for you guys to address. So if we switch over to exogenous factors, these are things that you can control, and especially when we talk about exercise, that's also the other knee-jerk. Well, that person just needs to do more exercise, a higher volume or a higher intensity or a different type, and those are all valid arguments, and I completely agree with those. Testing them gets a little bit more complicated. You can imagine doing a crossover of two different types of training in an individual will get complicated, but it still needs to be addressed nonetheless. And this question of time, asking someone, there was a recent paper that showed it was simply a matter of volume of exercise to convert these non-responders. And if you dig a little deeper in the paper, essentially what it took for these non-responders to see an improvement in their aerobic fitness, I think it equaled about 250 minutes a week. That's 100 more minutes than the physical activity guidelines recommend. So it almost becomes, it's not so feasible for everyone to be able to do 250 minutes of exercise a week. So I think we can do better. So, of course, I'm more focused on diabetics, and what we've seen in our own work and with, learning from other people's work is that these persistent training-induced improvements in insulin sensitivity were actually more dependent on training duration than the intensity itself, and that was with equal energy expenditure. And this was in a group of type 2 diabetics. But the duration mattered much more than intensity. Now, other people will argue that these volumes of exercise, and this was in a group of postmenopausal women, that had those individuals that had larger volumes of exercise. This was a dose-response trial called DREW from Tim Church, and they showed that the larger volume was associated with a lower probability of being a non-responder. So again, it's about time and volume is what we're seeing there. But then Marcus Bamman at University of Alabama in Birmingham studies resistance training almost exclusively, and what he found in a group of healthy folks, basically I think these were the college students, is that he could split up these college kids into extreme, modest, and essentially a non-responder to resistance training just based on the hypertrophic responses, so their ability to build muscle mass. And, it's a sort of offline discussion I've had with him that it always begs the question of, okay, well, just because these people can't build muscle mass, does that really matter for them? What if you put them in an aerobic training program? Would those non-responders still be a non-responder, or would they then move up to the extreme responder? It's a question that's, of course, unanswered, but it just gets back to these exogenous factors that we always have to consider whenever we're prescribing exercise intervention. What works for one might not work for another. So this phrase exercise resistance has kind of been thrown around. I don't use it as much. I think it kind of has a knee-jerk negative reaction, but nonetheless, it is still around in the literature. And it's just it's just looking at this, again, heterogeneity of the response, or these people that resist the response to the exercise training. So if we go back to the 80s, Claude Bouchard, world-famous geneticist, actually showed that the extent of the training response, he used a twin study, showed that it was largely heritable. So this was one of those first studies to suggest that your response to exercise could be driven by your genetics, this idea that athletes are born and not made, so to say. Or it could be an epigenetic modification. So since then, Larry Mandarino, I think he was actually here for some time. He's the one who actually coined this phrase, exercise resistance. So I stole it or borrowed it from him. And this was now 10 years ago. And he found in these subjects that were insulin resistant, they just did an acute bout of exercise, he found that they actually had this failure to increase expressions of these genes related to substrate metabolism, these genes that we know overall should be increasing in response to an exercise bout. Now, a little bit later, or sort of in parallel, a guy named Michael Roden in Dusseldorf has done some really nice work in individuals that are family history positive for diabetes or pre-diabetic or have diabetes, looking at taking this Claude Bouchard model and looking at their genetic predisposition for responding to exercise training, but focusing in on muscle mitochondrial function. So using this non-invasive measurement that some of you may be familiar with, but you have the capability here. It's 31-phosphorus. You get in the magnet, and I'll describe it a little bit later. But basically what he showed there is that a SNP in a single gene, a mitochondrial encoded gene for complex 1, actually, and I can show the data later on, it actually showed or predicted the response that these people had in terms of improving their muscle mitochondrial function. And I think that was 10 or 12 weeks of training. And Julian Zerath, getting back to this epigenetics, so it keeps sort of bubbling up to the surface. The genes can predict your response, and there might be modifications beyond that, sort of this epigenetic layer. And so what she showed, and this was in mice, but she showed that a single contraction actually induced DNA hypomethylation of promoter regions of genes that were involved in substrate metabolism. Again, these key genes that we know should be turning on or going up in response to a muscle contraction. So this sort of laid the groundwork, at least for me, in my mind, to apply for this grant to the ADA, which is all of the work that I'll present to you today, to sort of dig a little deeper, specifically in individuals with diabetes, trying to tease out some of the relationships between DNA methylation of genes in response to exercise training. And one part of that, we had some nice preliminary data that I had done during my postdoc off of the Heart-D study, where these individuals with type 2 diabetes exercised for 9 months. And this was supervised exercise. So this was an intense program. And we found that we took muscle biopsies, and just looking at their transcriptional profiles at baseline, So I should back up a second. We actually separated these folks into responders and non-responders based on their changes in HbA1c, body fat percentage, and muscle mitochondrial DNA content. And so we had some that responded, some that didn't. And if you just looked at, you can even look at a heat map that I'll show you in a minute, they differ dramatically just at baseline in gene expression of a lot of these genes were substrate metabolism related. So just at baseline, in a person's muscle tissue, you can already see differences in expression levels that is related to their response in terms of improving glycemic control and muscle mitochondrial content. That's not causal by any means. It's definitely correlative. But to be able to show that people are already unique from the start, I think that's where we can start to drill down and just better understand some of these things that could serve as a prediction. So what I care about, of course, is type 2 diabetes. And I've spoken to some of you about this, but I mean, I'm not an MD, I'm a PhD. But the current working model and the treatment paradigm for people with type 2 diabetes is, of course, they end up on this sort of spectrum. And, starting out with either family history of diabetes, there's this accumulation of adipose tissue, obesity, insulin resistance. You get some beta cell dysfunction. This can be reversible. But if you keep going, it moves into prediabetes. You ultimately get overt type 2 diabetes, beta cell apoptosis. That's irreversible, chronic hyperglycemia, and you ultimately end up reprogramming muscle stem cells. But the thing is, if you intervene at any one of these stages, you can sort of get people off the train tracks. But what we do now is it's diet and exercise as the first line of defense. Then we go to medication. And then we end up with people that are moving all the way down here before we say, hey, okay, let's put you on insulin now that you have no more beta cells. So I would like to start trying to shift this model where maybe we can find out what works for people, what doesn't work for people, and identify the ones that aren't responding to these exercise interventions, and then why are they not responding, identify these targets, and maybe you can have this beautiful combination of pharmacotherapy with exercise or pharmacotherapy with diet and exercise. That's up for discussion and left to discover. So I don't have to tell this room. It's diabetes is multifactorial. You get diabetes. If you have diabetes, you're a diabetic for lots of different reasons. It's not one flavor. They're not all the same. Some people have reduced muscle glucose uptake, beta cell failure, uncontrolled hepatic glucose production. Some have high rates of lipolysis. You can get ectopic lipid accumulation. So you get there for lots of different reasons. So it is going to be complicated and not easy to study. But here we go. It's going to be a wild ride for sure. It already has been. So I mentioned the Heart-D study, and if you're not familiar, this was a study done at Pennington by Tim Church. It was 262 individuals with diabetes, exercised for 9 months. They had a control group. And so I had the great fortune of being there at that time. And what I did was I got an ancillary study to essentially ask people to submit to a muscle biopsy and a blood draw. So took blood and muscle. They were randomized. That was the part I couldn't control, to one of these 3 groups. They were comparing aerobic versus resistance versus the combination. And then 9 months later, repeat that. So we ended up with 52 completers, 10 controls. And one of the things that we found that I thought was interesting is that the duration of diabetes, so how long these people had diabetes, and this is certainly imperfect because you can see there's this group up here that is not necessarily following the line. So duration of diabetes is not going to explain everything, but it was definitely related to their improvement in HbA1c. So anyone that, the longer they had diabetes, it sort of blunted their ability to improve their HbA1c with the exercise training. I think around the same time, John Kerwin published this paper where he had sort of gone back retrospectively into a lot of his data sets and found, he was finding that this ambient hyperglycemia was also blunting these people's response to exercise. So it's kind of a messy, busy graph, but essentially what he found is that for every millimole per liter increase in their pre-training baseline, 2-hour glucose that was above 13.1, they had a loss of improvement in response to exercise. So indicating that this hyperglycemic conditions can influence an individual's response to training. Again, it's messy. We got above and below, so hyperglycemia doesn't explain everything in everyone. Work by Barry Braun has sort of shed some light on metformin. It's obviously a fantastic anti-diabetic medication. It's usually the first go-to for most clinicians to prescribe to their patients. And granted, he's used sort of a hammer here with 2,000 milligrams a day. But what he did was, he's done this a few times. This is just one of his studies, either medication-naive, pre-diabetics, or a healthy population of individuals. He basically tested the effects of metformin versus exercise on insulin sensitivity. He did this by insulin clamps. And what he found, you can see, is that there is this blunted effect. So you have people, as we expect, as we've seen a million times in the literature, someone exercises, they improve insulin sensitivity. You give them metformin, and it blunts that response. Now, an obvious issue here is the black bars at baseline. Can't predict this. They were randomized, but the metformin group does have a much lower insulin sensitivity to start, so sort of a caveat to the study. But nonetheless, this data in combination with other data from other groups, a lot of in vitro work has shown that metformin in particular is a complex 1 and ATP synthase inhibitor. And so these biguanides are actually now classified as that. So you can imagine when your primary outcome is mitochondrial function, and especially insulin sensitivity, you need to keep that in mind, that people that are on metformin this might affect their ability to respond, at least in terms of that. Doesn't mean that they won't respond in other ways to the exercise. So this is that work by Michael Roden's group in Dusseldorf, where it's called in vivo ATP synthase rate. It's basically a non-invasive measurement of muscle mitochondrial function. And they were all family history positive, but you can see it pretty clearly here, dividing them into responders and non-responders, and this is just based on a single nucleotide polymorphism in one gene in these people. And that was enough to predict whether they would respond to exercise and improve muscle mitochondrial function or not. So then going back to this, as I called it, the Heart-D ancillary study, these folks that we took the muscle biopsies from before and following the 9 months of training, we did what we call these ex vivo metabolism assays, where we measure, basically take the muscle tissue, homogenize it, and we put a labeled fatty acid or pyruvate. You can pick lots of different things, but we used palmitate in this case. So you label it, that's this black circle, and you literally can measure the oxidation, the beta oxidation, because you're measuring it all. You're measuring or capturing the CO2 levels. So this is an indication of fat oxidation by the muscle tissue itself. So it's ex vivo, not in vivo, but as close as we can get to the in vivo in this case. And what we found here, so just kind of looking at the fold change after 9 months of training, and we reported this data that's great. Exercise increases muscle fat oxidation. But it's when you start looking at the data, you've got these individual data points, and it's been calculated in our data sets and other data sets that it ends up being on the order of about 15 to 20% of these people actually don't respond. So when you put that in the context of the 29 million people with diabetes, that becomes a little bit more impactful. So it's definitely worth investigating why these people are not responding and not just leaving them below our standard errors and saying, okay, everyone improved. I would like to understand why these people didn't improve. And so to put it in another context, this is their fat oxidation. It's these same folks, but this is just at the end of the 9 months. And these are lean, healthy individuals. You can see there's a huge spread, even amongst lean healthies. But the bottom line is you can actually train these diabetics, at least in terms of muscle fat oxidation, up to the level of young, lean, healthy folks. But again, you've still got this group down here that just isn't sort of making it over the hump. So we set out on our first adventure. I had a very talented postdoc, Natalie Stevens, who's moved on to a very nice industry job, but we were sad to see her go. But one of her first projects was to dig into this transcriptional profile of what a non-responder might look like. So going back to that Hart-D ancillary study, this is what I mentioned. We took these folks and looked at the measures that improved significantly overall for the group. So we knew that the exercise improved their HbA1c, we knew that it reduced their body fat, and we knew that it increased their muscle mitochondrial DNA. So that was in all of these 45 subjects we had. But some people did not respond. So using a principal components analysis, throwing all three of those endpoints into the model, what shakes out is you end up with these extremes, these 9 that were super responders, and then you end up with these 9 folks that didn't respond. And so this is just showing you basically what those numbers look like. Of course, I mean, it's set up to be that extreme, but you've got responders really improving over here, non-responders showing no improvement. And prior to exercise training, this is that heat map I was telling you about. So you just basically take their muscle tissue, looked at the RNA expression profiles, and I think this is about 200 genes that were significantly different. But you can see just at baseline, and we did ingenuity pathway analyses, enrichment analyses to see what the genes were related to. And not surprising, a lot of them probably also by design are related to substrate metabolism. So it's just tip of the iceberg, and this was some of the preliminary data we used in the grant. It's definitely not telling us that one particular gene is predicting this lack of a response. But one other thing that we noticed is, and I've talked to other people, just asked them to go back and look at their data, you could argue that this is some sort of regression to the mean, and I honestly don't know that I can necessarily answer that, but what we do find is this inverse relationship, at least with mitochondrial DNA copy number in muscle, as well as PGC1 alpha expression, that basically the higher, there's almost this ceiling effect, or a regression to the mean, if you want to call it that, where the higher you start out, the lack of a response, or they even decrease the expressions or the content of their mitochondria. And it's something that I still don't know that I can explain, but we continue to see this over and over. So I'm still trying to look for answers there. But it is suggesting that, the bottom line is that it suggests that some of these people are just unable to respond. Yes?
Question. So there are differences at baseline in muscle, transcriptionally and mitochondrial DNA copy number. Do you know about other tissues besides muscle? Do they differ between?
I would love to. Adipose tissue is another one of my favorites. I actually started in adipose tissue, but to be honest, I haven't looked at any other tissue. I've looked at just baseline clinical characteristics, sort of the low-hanging fruit, and there weren't any differences there. But as far as other tissues, no. And from this particular study, I only had muscle and not adipose. That's also the case for the data I'm going to show you in a minute. But I'm a believer that it's certainly not tissue-specific. I mean, muscle is not the only target of exercise. There's liver, gut microbiome. I know that's a popular one. Brain, all of it. Yes, sir.
Is this just diabetics, or is it all subjects?
This is just diabetics. Yes?
You were talking about a SNP, and you said it was a mitochondrial gene. Is it a nuclear gene, or is it mitochondrial gene?
Mitochondrial encoded. It's a mitochondrial encoded mitochondrial gene. It's INDUF6. It's part of complex 1.
Okay. So that's going to have something to do with the number of copies, mitochondrial DNA.
Right, and so the mitochondrial DNA. So that was the SNP was from another study, not from work that I've done. And there, they didn't look at mitochondrial DNA. So the mitochondrial DNA is from my study, but I have not looked at. I don't do mitochondrial genetics, but I have a colleague that's delving into it. Actually, as part of the MotorPack application, they're wanting to look at mitochondrial genetics for lots of these reasons, because we know that the content versus function is always an argument that people have in this field. So far I've showed you essentially all the data and the reasoning around, my reasoning at least, around wanting to look at response heterogeneity in type 2 diabetics in terms of exercise. So it formed these hypotheses and rationale for doing this study, kindly funded by the ADA. And the bottom line is that exercise benefits most, but not all individuals with type 2 diabetes, at least in terms of glycemic control, mitochondrial function. But it hasn't really been well studied, in fact, hardly studied at all. And so the goal of this was to start to unravel, just to touch on maybe some of these mechanisms that could be underlying it, in particular DNA methylation. So the overarching hypothesis is that there would be this deleterious epigenetic signature, but let's be honest, epigenetics is quite broad. This is DNA methylation in the skeletal muscle that's preventing this beneficial effect in response to exercise training in terms of muscle mitochondrial function. So the approach had two steps. I'm clinical and translational, so I tend to start with a clinical intervention. So the idea was to identify these responders, non-responders. You have to set your outcome at the onset. So identifying these people based on their response to the exercise intervention for in vivo muscle mitochondrial function, and then test this hypothesis that there is this pre-training epigenetic signature that would be related to that response and lack of a response. And doing that, as I said, in vivo, and then we did muscle biopsies and cultured the muscle cells, so these satellite cells or myogenic progenitor cells, to see if that's preserved in vitro using DNA methylation and RNA-seq. So just a pretty representation of what I'm talking about. I know some of you are familiar with the Ouroboros, but I'll be introducing that as well. With the addition of a new postdoc to the team, Dr. Braun-Browers took over a lot of the work once Dr. Stevens moved on. And just a general design overview, we ended up with 3 groups. We had 17 type 2 diabetics that completed. We had a group of age and BMI matched non-diabetics. And then we had this group of highly active individuals. I'm not going to call them athletes, but basically a VO2 max over 45. And they were doing at least 2 hours of exercise a day, more than 4 days a week. So highly active. They essentially, the first 2 groups underwent metabolic assessments at the beginning, did 10 weeks of endurance training, essentially walking on a treadmill, and then repeated those assessments at the end. One important point, because as I pointed out, this issue with metformin and the exercise response is we set out to have medication-naive diabetics. That ended up being almost impossible to recruit. So we were willing to take folks that were on 1 or 2 medications that we could wash off medication for 2 weeks using continuous glucose monitoring for safety, and then we were going to intervene. So at least from a safety perspective, we were intervening while they were off their medication. And then with the idea being at the end, this in vivo mitochondrial function, I'm now going to refer to it as PCr Recovery Rate, and separating the groups into responders, non-responders that way. So this in vivo, this PCr Recovery Rate is measured in a 3 Tesla magnet. Like I said, you guys have one here. And the folks basically go into the magnet. They'll do knee flexion and extension. And in real time, we're measuring the decline in PCR and then the recovery. And I'll show you the actual data in a second. They did a VO2 max test. They had a muscle biopsy followed by an insulin clamp and then underwent the intervention. So why select this response variable? I mean, there are lots of tissues, as she was just asking. There are lots of tissues that are affected by exercise. And then even within muscle, there are lots of things you could choose. But because I care about this relationship between mitochondrial function and insulin sensitivity, and that's certainly an important clinical outcome for people with type 2 diabetes, we chose in vivo mitochondrial function. And this is work from my now boss, previous mentor, showing that there is a decline when you go from highly active to individuals with diabetes. There's a decline in this PCr recovery. But, of course, there are some diabetics that are right here in the group with some healthies, and then there's some down here sort of on their own. So there's quite a span. And And, basically a mirror image of this graph, you see the same thing going with insulin sensitivity going all the way down. So suggesting a link, but certainly not. It's argued still in the literature, and so they're linked, but cause and consequence, who knows? Depends on who you talk to. So looking at this variable, Dr. Heather Cornell is our resident physicist who's much smarter than I am and actually understands this measurement or all the ins and outs. So as I said, it's just a brief period of knee flexion and extension while they're in the magnet against a resistance band. And because there's minimal energy demand at rest, we're putting this stress on them, or basically putting an ATP demand. So that's why people refer to this as this maximal capacity to synthesize ATP. And this is just an example of the spectrum, but basically what we're measuring is this peak here, the PCr, and its decline. And we also want to make sure that these folks don't get acidic, so we don't have contribution from glycolysis. So we're able to measure, after the fact, we go back and measure pH, and if anything, we get them to about a 50% decline because we know that that typically avoids a drop in pH, but of course, whenever we look back and if we have a drop in pH, we get them to repeat it. So something that's very important in picking these outcome variables is that you have this sort of test, retest, reproducibility, because you want to make sure that you're not just measuring an artifact or an error of the measurement itself. So what we've done when we first set up the method was so this is them at rest, they start kicking, and like I said, you're watching in real time the decline in PCr. We tell them to stop kicking, they're at about 50%, and then they just lie there and we measure the recovery. So our CV is 4.5%, which, I mean, from what we found out from other places that are doing this, I'm not sure what your CV is here, but this is about where we want to be. I'm not sure that I've seen much better. So these are some of those papers, and actually I think, no, I don't have the one up here that I was referring to, but this is just more data showing, not just from us, that these myotubes retain the metabolic phenotype. This is illustrating metabolic flexibility. So if you titrate in glucose and measure fat oxidation, it's this suppressibility, this ability, Randall cycle ability of glucose to suppress fat oxidation, and then sort of the reverse, this adaptability to increasing amounts of fat as it's titrated in. So this is termed the in vitro metabolic flexibility, and they showed that here it positively associated with delta RQ, or the in vivo metabolic flexibility during a clamp, and also with insulin sensitivity. So we took these cells just at baseline, not post-training, so similar to what I showed earlier, but that was muscle tissue from that other study. So these are now the cells at baseline from responders and non-responders. I'm just giving a table here just to kind of keep it in context and remind everyone of their the changes in PCr recovery, insulin sensitivity, and A1C. And I think it pretty much speaks for itself. We had almost 500 differentially methylated CpG sites, and you can see a stark contrast between responders and non-responders. Shift this paradigm, this current treatment paradigm that we have for diabetes, and maybe level the playing field. Maybe you wipe these people clean of hyperglycemia. You can't wipe away things like family history and people's genetics, but if you can even the playing field, get these diabetics maybe into remission, and then intervene with exercise, you can sort of disentangle where these breakpoints are, and try and combine in a better way, better than we're doing now, which is just blanketly prescribing, just go exercise. Physical activity guidelines say do 150 minutes a week, and then think it's going to work for everyone. So ACSM says exercise is medicine. I say maybe sometimes. But I'm a proponent of exercise, and this is my 2-year-old daughter, and she runs races as much as a 2-year-old can run in a race, but I am a huge fan of exercise. I think we can do better. And so, of course, with every study, there are limitations. This is a small cohort. As I've mentioned, we did wash these individuals off medication, so we induced hyperglycemia, which has profound effects on epigenetics and lots of other things. And it was only endurance training at a moderate intensity, walking on a treadmill. But I like to think that we have opportunities, especially when you think about, like I said, all of these people experienced a medication washout. So if we can start them off all at the same point and then try to disentangle what the regulation is of their response, I think we'll be doing a lot better. And so you could begin to identify genes here that could be tied into these larger studies that are going on, which is, you know, the NIH Common Fund. I'm sure you guys have heard. Dr. Mussi is the PI. It's a big deal. The director of the NIH thought that this was a big enough deal to understand why exercise is good. We know exercise is good, but why? And what are those? Essentially, MotorPAC stands for molecular transducers of physical activity. And understanding or developing this temporal map in response to exercise bouts as well as a training intervention in sedentary people, I think once we have those maps, at least in muscle and adipose tissue, we can start to take studies like this that have these smaller cohorts in diseased populations and sort of lay those against the maps that will be developed from MotorPAC. So these are just a quick overview, the sites of MotorPAC, and you can see you guys, you know, we're on here in Florida. You guys are here, and we are. So the aim, I already told you, is to develop this comprehensive map of molecular changes, but more importantly, we are also joining forces to put in a couple of ancillary studies to MotorPAC, and not surprising, I am wanting to establish a repository of these myogenic progenitor cells and also do some functional analyses, and I've asked Dr. Mussi to also be a part of that, and also not surprising, this in vivo mitochondrial function leverages your ability to quantify in vivo muscle mitochondrial function with your beautiful 31 phosphorus MRS measurement. So hopefully we get lucky and land at least one of those. And yeah, I'll just close by, as you know, clinical studies are beasts, and it's never one person, and it takes a lot of brilliant people that are willing to participate. So I just want to thank, of course, my mentors and postdocs and all the bioinformatics folks that have dedicated lots of hours to this. So I'll take questions. We've had questions throughout, but I don't know how I did on time.